Hello, good morning everyone. Today we will study and understand what is respiratory membrane and what are the principles, physical principle that is governing the exchange of gases between the alveoli and the capillary of the alveoli that is being supplied. So, what is a respiratory membrane? So, this respiratory membrane is what we are going to understand first. Now, talking about the function of the respiratory membrane. What is the respiration? Respiration is happening between two structures. So, two structures in the whole respiratory system that are involved in gaseous exchange. So the two structures are, first one is the alveoli and second one is the capillaries, the pulmonary capillaries. So when this exchange of gas is taking place between this alveoli and the capillary, there is a specialized structure which is kind of modified or adopted so that this gaseous exchange can take place. Because when we are breathing in, we are breathing in from the atmosphere. From the atmosphere, it comes all the way to the lungs and it goes to the alveoli, which is the unit of respiration. So, this is the gas part of it. And how does the blood part get connected? It is via the capillaries that the blood is coming in contact to the alveoli. Now, the specialized structure by diagram, let us look at it. So, the respiratory membrane, to understand in a simplified way, we draw it in a box as per the layer for our understanding purpose. So, these are nothing but like a portion of the alveoli. Say this is like the layer of the alveoli, which is in contact with the layer of the endothelial layer of the capillary, because we know capillary is made up of only one layer of endothelial lining. So the layers in the respiratory membranes are like in multiples. The first structure that is involved in the respiratory membrane is like a layer of surfactant. So we know in the alveoli there is a coating of surfactant, and surfactant alone is not there. It is also having a coating of the fluid. So this is our layer 1, which is on the alveoli side. Now interior to that, what we have interior to that is the second layer, which is the epithelial layer of the alveoli. Alveoli, as we studied earlier, it is made up of a simple epithelial cell. So that one layer is there. Now, after that, what comes is a basement membrane. Is a layer of, like, there's a layer of basement membrane. Basement membrane on the which side? On the alveoli side. After that, there are like layers of this interstitial fluid. Like there are this layers of this interstitial fluid which is adjoining or connecting to the endothelial layer. So, the fourth layer is the layer of interstitial fluid. Now, via this interstitial fluid, actually there is like contact between the alveoli as well as the capillaries. Now on the capillary side starts the layer related to the endothelial lining of the capillary, that is after the layer of the interstitial fluid. So now what is next to it? It is the fifth layer, which is the basement membrane of the endothelial layer of capillaries. And lastly, what we have lastly is the endothelial layer or the capillary endothelial layer. Now, this 6 layers that we have studied, this is layer number 1, then comes the layer 2, 3, 4, 5, 6. So, this 6 layers actually makes up the respiratory membrane, means it is via this layer, like this is the alveoli side and this is the capillary side. So, when we are breathing in air from the atmosphere, atmospheric air is having more of oxygen concentration. So, what happens is this, there is a direct diffusion exchange of oxygen from the alveolar layer across the capillary, because it is from the capillary that the oxygen will bind in the blood and then go back into the circulation, into the systemic circulation via the heart, that we have studied in the cardiac cycle. And the carbon dioxide that has to be eliminated out, that is also happening across this respiratory membrane via the alveoli and then it is expired out to the atmosphere. This is the respiratory membrane. Now the surface area of the respiratory membrane is about 70 meters square. That's how big is the surface area across which the exchange of these gases take place. And its thickness, its thickness is about 0.2 to 0.5 micrometer. That's how the thickness is. Now, this is the structure across which the exchange of gases takes place, that is the respiration takes place. Now, there are also certain criteria, certain law that carries out this exchange of gases. Now, gas, we are inspiring and expiring gas, and gas, whenever it has to move from one place to another, the principle, the law it follows is always the physical principle, because it is gaseous in nature. So, what happens is that the principle for diffusion of gases across the respiratory membrane, respiratory membrane follows the Fick's law of diffusion. So, it is the Fick's law of diffusion that carries out the exchange of gases. So, now first we will understand the Fick's law of diffusion and the factors controlling it. Then we will go on to understanding the laws governing this Fick's law, like the laws of the gas. Now, Fick's law of diffusion, it says that diffusion of gases, which is designated as V gas, is a function the direct function of the surface area of the respiratory membrane Meaning what? Meaning that here, if the surface area of the respiratory membrane is bigger, larger it is, say in a person, the respiratory membrane, the surface area is 30 meters square, and in this another person of the same age, same height, same stature, the surface area is 50 meters square. So obviously, just an example. So obviously, the rate of diffusion of gases in the person which is having 50 meters square of the respiratory membrane will always be bigger, will always be faster, will always be more. So this is a direct function. Second is the diffusion coefficient. Now, diffusion coefficient, it is a calculated standard thing that we already have a value for. Like for example, the diffusion coefficient of oxygen is 1. It is a calculated one, the established one. Whereas the diffusion coefficient of carbon dioxide is about 20. It's 20.3 something, like roughly we are taking it as 20. So here carbon dioxide is greater by 20 times than oxygen in diffusion. That means if same concentration of carbon dioxide and oxygen are present, the carbon dioxide will diffuse readily. It will diffuse at a much faster speed compared to that of the oxygen, because the diffusion coefficient of the oxygen is 20 times lesser, or rather carbon dioxide is 20 times more than that of the oxygen. So diffusion coefficient plays an important role. Another one, that P, as we see here. So P is the partial pressure of gases. Let's talk about it. Now, partial pressure of gases. Okay. So, in the atmosphere is a mixture of gases. It has got nitrogen, it has got oxygen, it has got carbon dioxide, monoxide, and also, miscellaneous gases like H2S, ammonia, but in very less quantity. The maximum amount of gas that we have in the atmosphere is that of nitrogen. Then we have oxygen also. Now, the oxygen, partial pressure of oxygen at the sea level that we are inspiring in is much more than that of the carbon dioxide. Nitrogen doesn't count because nitrogen is not, nitrogen is not taking part in the process of respiration. So, nitrogen doesn't count. So, here, like the atmospheric partial pressure of oxygen, it is about, at sea level, it is about 100 mmHg. Okay. Whereas the capillary, the capillary partial pressure of oxygen that is carrying to the alveoli, because that capillary blood that is carrying the oxygen to the alveoli, it is rich in carbon dioxide, it is less in oxygen. So that partial pressure is 40 mmHg. So you can see here by difference, when from the atmosphere we are inspiring the air, it has a partial pressure of 100 mmHg, right? That is the one that is coming through the alveoli, coming through this structure. Okay. Now, when it is coming in context to the capillary, now this capillary is having a partial pressure of oxygen which is only 40, So now, if this atmospheric pressure, if we designate it as P1, and that in the capillary, if we designate it as P2, so P is equal to P1 minus P2. So here you see 100 minus 40, obviously mmHg and there is a difference of 60 mmHg. So you see the oxygen from the atmosphere, since it is high on partial pressure, it is having more capacity to diffuse easily because its concentration is much more than the concentration of the oxygen that is in the capillary. So partial pressure plays an important role. So what is the total take from it? All this partial pressure talks that I have done now, what exactly we have to remember? We have to remember is that if the partial pressure difference, again following the diffusion principle, if the concentration gradient, if the difference in concentration is more, then the rate of diffusion will be higher. So these three factors, surface area, diffusion coefficient and the partial pressure, these are directly related to the diffusing capacity according to the Fick's principle. Whereas the fourth factor, that is the T. So here T is the thickness of the membrane. Obviously the respiratory membrane, we know the normal thickness is about 0.2 to 0.5 micrometer. That is the normal range of it, value of it. So if this thickness is more, then obviously the gas will have more difficulty in passing, in getting exchange. So this thickness has an inverse relation. If the thickness is more, then the diffusing capacity will be less. So this is what is the principle of the diffusion of gas following the Fick's law. Now, clinically, how do we correlate to this one? What is the applied part of it? Let's talk about the applied aspect of the Fick's law of diffusion. Okay, what is the aspect part of it? Like, say, number one, we start with the surface membrane. So, in people with emphysema, we know what is emphysema, where the elastic recoil is lost. In emphysema, the surface area decreases. So, the rate of diffusion also decreases. Okay. So, people having problem, having like conditions like the COPD, emphysema, in them the rate of diffusion is low because the surface area becomes less. Another factor, like in people with fibrosis of the lungs, basically where the scarring of the lung tissue happens. So here in the fibrosis of the lung, the thickness of the respiratory membrane increases and rate of diffusion decreases. Okay, these are the applied aspects of the Fick's law of diffusion. Now, this is about the respiratory membrane. Now, what are the law that governs the diffusion? So, respiration and the respiratory membrane being connected to the gaseous exchange. There is more than one principle of the gas that carries out this respiration, this exchange of gases. So, laws governing exchange of gases. The first law that we need to know is the Dalton's law. Now, what does Dalton law talks? Dalton law talks about partial pressure. According to it, the total pressure exerted by the air is sum of the partial pressures of many gases. Okay. Suppose if P is the total pressure, if it is the total pressure, then P is equal to P1 plus P2 plus P3, where P1 may be the partial pressure of oxygen, P2 is a partial pressure of carbon dioxide, P3 is a partial pressure of carbon monoxide, like that. So it is the sum of the multiple pressure that is determining the total pressure exerted by the atmospheric air. Second law, in context to that, is the Henry's law. Now, Henry's law, it talks about the relation between the concentration of the gas, partial pressure and solubility. Like according to the Henry's law, N is equal to P into C, where N is the concentration of a gas, P is the partial pressure of a gas, and C is the solubility of a gas. So, the concentration of a gas will be more if the P, the partial pressure and the solubility coefficient is more. Like in context to that, as we have already studied, carbon dioxide is 20 times more soluble than oxygen, and carbon monoxide is 200 times more diffusible than oxygen. Okay, these are the laws that are governing. Now, how do we apply this law in understanding the concept related to the clinic? So, it is like that we will understand with the graph. So, looking at this graph. Okay. So, we are going to zoom out the graph a bit. Okay. Now, when we are inspiring gas, there are more than one gas, as we know, that gets into the respiratory tract. Now, the primary gas, the major gas are the nitrogen, which is an inert gas, the oxygen, the carbon dioxide. And carbon monoxide also gets inside the system, but altogether it follows a different mechanism. Now, looking at the graph, we can understand this is a graph that is denoting on the x-axis. If you see, it is denoting the time that the gas takes to enter through a capillary and leave a capillary that is surrounding the alveoli. And on the y-axis, we have the partial pressure of the alveolar gases. Now, these gases that are present in the air, not all of them are dependent on diffusion. Some gases are such that they are controlled by perfusion. Like, what do we mean by that? This is a very crucial concept which needs to be clearly understood. Like gases like oxygen and nitrous oxide are perfusion dependent. Means what? Perfusion means what? Perfusion means the blood supply. So it means that if the concentration of oxygen and nitrogen is more in the blood, or if the blood flow to the alveoli is more in case of hypervolemia, suppose there is a condition when the blood flow is more, then the gases like oxygen and nitrous oxide will get exchanged across the alveoli more. Whereas carbon dioxide carbon dioxide, or rather most important, carbon monoxide is diffusion limited. Now, what is diffusion limited? This carbon monoxide, its concentration will increase only if its rate of diffusion is more. So 2 points we learned, then we are going to go details into it, that gases like oxygen and nitrous oxide, they depend on the blood flow. Means if the blood flow increases, the concentration of the oxygen and nitrous oxide getting exchanged across the alveoli will be more. They are not dependent on diffusion. They are not dependent on diffusion. But carbon monoxide, it has got nothing to do with the blood flow. It is dependent only via the diffusion. So here you see, if we look at the oxygen graph as highlighted here. So when the oxygen is entering via the capillary, now usually the transit time is like 0.7 seconds. Okay, it is 0.7 seconds. Like the blood, it takes about 0.7 seconds to enter the capillary and leave the capillary. So oxygen, when it is entering the capillary, it becomes like stable. The diffusion stability or the diffusion equilibrium is attained within 0.25 second to zero point 0.3 second. So just within 0.25 second, the equilibrium, means the partial pressure concentration between the alveoli and capillary is attained. So, diffusion does not control the oxygen that is getting exchanged. If the blood flow increases, if the perfusion increases, then more blood means more oxygen coming to the lungs, more oxygen coming to the alveoli. So, the capillary will be carrying more oxygen. This is what it means. Same nitrous oxide also, if you look at the graph, it is attaining equilibrium at a period of like less than 0.2 second. These two are perfusion limited gases in normal condition. But whereas if we look at the carbon monoxide, what we see, carbon monoxide, it is not dependent on perfusion. It never attains equilibrium with the alveolar pressure. If the rate of diffusion is more, its concentration is more. The reason for that is that, why it is so? The reason for that is that oxygen, carbon dioxide, they can dissolve in plasma. Okay. But carbon monoxide cannot get dissolved in plasma. Then how do they get transported? Carbon monoxide, point to remember, carbon monoxide, it can only replace the oxygen from hemoglobin and bind on the hemoglobin. That's why only it is diffusion related. It does not get dissolved in the plasma. And for partial pressure to be exerted or for the equilibrium to be attained, dissolving in plasma is essential. Now, in normal condition, we have seen that oxygen is like limited by perfusion. But in cases of emphysema and fibrosis, the oxygen transport become diffusion limited. Now, when there is any defect of the respiratory membrane, like in case of emphysema, the surface area becomes small. In case of the fibrosis of the lung, the membrane thickness becomes more because of the fibrosing, because of the scarring. So, in that cases, the diffusion equilibrium or the equilibrium that is attained between the capillary oxygen and the alveolar oxygen, it becomes independent of perfusion, because even if the blood flow is normal, due to fibrosis and emphysema, proper exchange cannot take place because the membrane itself is defective. So it cannot follow the Fick's law. In that condition, in that time, it becomes dependent on diffusion, and the same graph will look like this. The same graph will look like this thing. It will become abnormal and it is changed to this. So this is what on our top we have the solid line, the continuous, this O2, this is the one that is in normal condition. But when the oxygen becomes like diffusion related, limited, not the perfusion limited, that time the same equilibrium graph, it shifts below, and instead of attaining a hyperbolic shape, it becomes like kind of like shortened, pushed towards downward, and it attains a gradually slow like structure. This is the abnormal diffusion of the oxygen while transiting through the capillary, and this is seen in patient who are suffering or people who are suffering from fibrosis of the lung or emphysema. Okay, this is in context to the respiratory system. Coming to the content of the oxygen. What are the partial pressure? What are the content of the oxygen that is being carried in the blood? So, oxygen in blood is studied as content in the arterial blood and content in the venous blood. Now, theoretically, when the partial pressure of oxygen, is 120 mmHg, it is called 100% saturated. So when the partial pressure of oxygen is exerting a pressure of 120 mmHg, it is called to be 100% saturated. Now, in the atmosphere, oxygen percentage is about 21%. So, in atmosphere, content in atmosphere is 21% into 760, because 760 is atmospheric pressure, which is about 160 mmHg. This is how much of oxygen is present in the atmosphere, provided the atmosphere is like clean. It is not in a polluted condition. Okay. Now, the same partial pressure of oxygen in alveolar air or alveoli is about 100 mmHg. So here you see, suppose even if we are inspiring 160 mmHg of oxygen, suppose, though it's not 160 mm of Hg because of the pollution and everything. So say even we are inspiring 120 mmHg of oxygen from the atmosphere, by the time it goes to the alveoli, it is only 100 mmHg, which is about 100% saturated, not totally 100%, which is almost 100% saturated. Okay. Now, why does it gets like dissipated? Because it is passing through the conducting zone. It is passing through the respiratory structures that is made up of cartilage. It has got epithelial lining, though it has mucus also. It is air. So, during the friction, some oxygen is just lost. Okay, which is not even taking part in the respiration. By the time oxygen reaches alveoli, its partial pressure is 100 mmHg. Now, the same partial pressure of oxygen in the arterial blood, like after the gaseous exchange has taken place and the arteries has the freshly bound oxygen, then also the partial pressure drops by another 4 to 5. It's about 95 mmHg. This how much is the arterial partial pressure of the oxygen. And now the partial pressure of oxygen in venous blood means the blood that is coming through the veins from the tissues after the metabolic reaction has taken place, after the cellular respiration has taken place, the blood that is carried back from the cells, from the tissues, that blood, it has about a partial pressure of oxygen 40 mmHg, naturally, because lot of oxygen gets used when there is the act of cellular metabolism, right? And also the partial pressure of oxygen in interstitial fluid, this is also 40 mmHg. These are the overall partial pressure. Now when we go on to calculating the total amount of oxygen in the arterial blood and in the venous blood, what we do, we take into account in what form the oxygen is present in the blood. Oxygen binds only to the blood. There is no other substance, no other structure, no other media in which the oxygen binds. So the only thing where oxygen binds and stay is in the blood. Now in the blood, how does the oxygen stay? So in blood, oxygen stays in two forms. What are the two forms? First, it stays as dissolved form. Now, if it has to stay in dissolved form, where it has to stay? It has to stay in the plasma, because plasma has 90% of the water, and then it has got the other things like, the other plasma proteins and all. But maximum what it has is water. So, in the plasma, oxygen stays in dissolved form, whether it is the arterial blood, whether it is the venous blood. In both the thing, it stays in the dissolved form. This is the first form. Second form, it stays is like bound to the hemoglobin. It also binds to the hemoglobin. Now, just a small talk on the binding of the or binding onto the hemoglobin. So, this point, let's talk a little bit about it. Now, in hemoglobin, oxygen binds reversibly. Okay. So it binds reversibly means what? Say a totally deoxygenated hemoglobin, it will bind with oxygen reversibly and form HbO2. So one molecule, that is two atoms of oxygen is bound. Now to that another two atoms or one molecule will bind and it will become HbO4. Now to that, like that, another will bind and it will become HbO6. Again, to that another molecule binds, and it becomes HbO8, which is a fully saturated hemoglobin. So, with one or each molecule of hemoglobin, totally 4 molecules of oxygen or 8 atoms of oxygen can bind. And this binding is reversible. This binding is reversible, not irreversible, means it can bind and also come out. So, the total content of the oxygen, that way, if we talk about, is the summation of the dissolved part as well as the bound part. So, how do we calculate that one? Say, in arterial blood, what is the content? It is the how much percent of oxygen is saturated. Now, the percent of oxygen that is saturated is about 95%. So calculating like 95% saturated hemoglobin. Now here also there is one thing that we need to understand. So when we talk about saturation, is that 1 gram of hemoglobin can carry about, not exactly, but about 1.34 ml of oxygen. So assuming that a person has 15, that is the international standard, 15 gram per deciliter of hemoglobin. So a person having 15 gram of hemoglobin can carry 15 into 1.34 ml, which is about 20 ml of oxygen. This is how much oxygen in fully saturated condition can carry oxygen. So 1 gram of hemoglobin carries 1.3 ml. So assuming a person has 15 gram of hemoglobin, so onto that 15 gram of hemoglobin, what is the carrying capacity? That 15 gram of hemoglobin per deciliter of blood can carry 20 ml of oxygen. This is the saturation point. This is the saturation point where in the arterial blood, So in the arterial blood, if we calculate, so it will be like, it is like about the oxygen saturation plus the dissolved oxygen. Now, how is the dissolved oxygen calculated? Dissolved oxygen is calculated by the partial pressure into the constant, that is 0.003. What is the 003? This 003 has come after calculating the oxygen partial pressure. So for us, it is enough if we remember this 95. It is a 95% saturated in the arterial blood and its constant 0.003, which is about 0.28 ml, assuming it is 95% saturated. And here the oxygen saturation in the arterial blood, assuming it is 95% saturated, not 100% saturated as here. It is about 19.5 ml. So in the arterial blood, the total amount of oxygen, that is there is 19.8 ml of oxygen. Where it is there? It is in the arterial blood. Whereas following the same calculation in venous blood, it is 15.2, because in venous blood the saturation is much less. It is 75% saturated, and here it is like 40 into 0.003, because 40 is the partial pressure. In the venous blood, the partial pressure drops to 40. So both this together is about like 15 point, it is 15.1. So it is about 15.3 roughly. 3 roughly. So this is the amount of the oxygen that is being carried out by the venous blood and the arterial blood. Okay, so these are the things. Now look at the values. Venous blood have about 15.3 to 15.2. So for our understanding benefit, let us make it 15.2. Whereas the arterial blood, it is carrying about 19.8 ml. So we know oxygen is a gas that flows following the law of diffusion. So following the law of diffusion, the arterial blood oxygen content minus the venous blood oxygen content is equivalent to 19.8 ml minus 15.2 ml. So for our convenience, let us round it up and write 20 ml because it's almost 20 minus 15 ml because it is 0.2 means less than 0.5. So what we get is it is roughly 5 ml. Exactly it is 4.6, but 4.6 is also equal to 5 ml. So what is the take from it? That the arterial blood, when it is going to the venous blood, when it is going to the venous blood. So arterial blood, how much oxygen was it carrying? Exactly about 20 ml. 19.8 for our understanding, we round it up as 20. So 20 ml. And when the venous blood is coming back for venous return, via venous return, it will go to the lungs, right? For purification, for exchange of gas. How much oxygen it is carrying? It is carrying like 15 ml of oxygen. 15 ml of oxygen per 100 ml of blood. So what does it mean? That 5 ml of oxygen is being given out, to the tissue from the arterial blood to the tissue, and 5 ml of oxygen per 100 ml of blood is being used by the tissues for its metabolic process, for its own cellular function, right? So every time 100 ml of blood goes to the tissue, it is using, or it is like giving away 5 ml of oxygen to the tissues. So to understand that, there is a measurement which we call as the utilization coefficient. Now, utilization coefficient, what does it say? Utilization coefficient, it says that oxygen, it is a total amount of oxygen utilized by oxygen produced, and it is in 100. So, its value stands out to be 25%. 250 by 1000 into 100, it value stands out to be 25%. So the utilization coefficient of oxygen is about 25%. Okay, this is the take from it. So what we have to remember, every time 100 ml of blood, purified blood, oxygenated blood goes to the tissue, it is giving out 5 ml of oxygen to the tissues for its cellular activities, for its metabolic purpose. Okay. Now, apart from this thing, there is also water vapor that we are inhaling. Now, the atmospheric pressure, what is the content of the water vapor? The content of water vapor in terms of partial pressure is 447 mmHg. Content of water vapor partial pressure. This is about 47 mmHg. This is something we have to remember. Now, by the time the alveolar air gets into the alveoli, means from the atmosphere, when we are inspiring air, the oxygen that is getting inside the alveoli, it again gets mixed up because it is a mixture of gases. So accordingly, the partial pressure and the amount also decreases. Now, there is a formula to measure that alveolar air depending on the partial pressure. So the alveolar, which sometimes you may have to know, you may have to answer. So alveolar air content, like depending on partial pressure, the content can be measured by the following formula, which is like partial pressure of the barometer minus the partial pressure of the water. Water is water vapor, that is 47 mmHg, into the fraction of inspired air. The fraction of the inspired air that we are inspiring minus the partial pressure of the alveolar carbon dioxide by the respiratory quotient. Respiratory quotient. Now, respiratory quotient means it is the amount of the carbon dioxide given out compared to the amount of the oxygen that we take in. That is the respiratory quotient, right? Now, this is what will give us the partial pressure of the alveolar air. This formula we have to remember because sometimes it may be asked in the exams. So, this is about the laws governing the partial pressure and the exchange of the gases. Now, we move on to the oxygen hemoglobin binding or dissociation curve, and what is the Bohr effect and the Haldane effect, which is of the clinical importance. Now, binding of, what is the concept on which the oxygen binds onto the hemoglobin? Binding of oxygen to hemoglobin. First thing, oxygen binds on to hemoglobin by positive cooperativity. And the binding of this oxygen to hemoglobin, if it is depicted in a curve, the curve is sigmoid in nature, and the sigmoid curve has got its own significances, its own importance. So, what is the sigmoid curve and its part, and what is it important? What is the importance of the sigmoid curve? So, the sigmoid curve, which we famously call as the oxygen hemoglobin dissociation curve. So, this is a curve which represent the partial pressure of oxygen on the x-axis and the hemoglobin percentage on the y-axis. So, below we can see the partial pressure written as 25, 50, 75, 100 like that. And on the y-axis scale, we have the percentage of saturation. So, here you see, look at the nature of the curve. The nature of the curve, it has got two parts. The curve has a flat portion. It has a flat portion. This one, which is like the flat portion, and then it has a steep portion. It also has a up climbing. There is a steep portion in the curve also. There's a flat portion and then there is a steep portion also. Now, the flat portion where it has leveled off, which is like marked with the arterial blood. Now, this is the maximum saturation that the partial pressure of oxygen has in the arterial blood, which we can see to be almost about 100 mmHg, though it's like it varies from 95 to 98. So, here it is shown as 100% saturated. Okay. And here, if we look at the steep portion, what is the significance of the significance of the flat portion first? The significance of the flat portion is that here the oxygen hemoglobin affinity is maximum, is like highest, means the affinity, the strength, the bond with which the oxygen is binding on the hemoglobin, it is maximum because all the four, at this maximum point, all the four molecules or 8 atom of oxygen has bound on to the hemoglobin. So the affinity, the strength with which the oxygen has sat on the hemoglobin, it is maximum. It is very tightly, very strongly bound. And at this partial pressure, that is at 100 mmHg, like at a partial pressure of 100 mmHg, where the saturation is 100% saturation, in this condition, it is very difficult. In this condition, Here, the unbounding of oxygen is very difficult at that point. This is the first significance. Next, another thing to be noted is that if we see very finely the partial pressure, like at 100 it is maximum. Now when the partial pressure from 100 it drops to 75, even at a partial pressure of 75, still if we draw a line that goes and vertically it joins the graph. So here also we see when the partial pressure drops from 100 to 75, still the oxygen hemoglobin association affinity binding is very, very strong. Means there is no detaching of the oxygen from the hemoglobin. So the second significance is that even when the partial pressure of oxygen drops to 75 mmHg, still the oxygen hemoglobin affinity is strong, is very, very strong. So, with a decrease in the partial pressure from 100 mmHg till 75 mmHg, there is no opening up or there is no unloading or unbounding of the oxygen from the hemoglobin. That is the significance of the flat portion or flat part of the sigmoid curve. Now, when the pressure drops further below, like from 75 it starts dropping to 50, and from 50, like if we can draw a vertical line and connect it to the sigmoid, so 50 and below, if we see 50 and below, like from 50 it drops to like 40, 30, like that, when it starts dropping we see the steep portion. Steep portion means here the oxygen and the hemoglobin affinity decreases. So now significance, the significance of the steep portion of the sigmoid curve. So what is the significance of the steep portion? When the partial pressure of oxygen is 50 and below, the oxygen hemoglobin affinity decreases. It starts to decrease. And when it starts to decrease, oxygen starts coming away from hemoglobin and is available to the tissues. Okay. So at a partial pressure of 50 and below, like when it is 40, 45, 35, like that, here we see the steep portion, means the oxygen is getting dissociated from the hemoglobin. So when the partial pressure starts dropping, that means the tissues, the cells, they need more oxygen, because cells, they cannot survive without oxygen. The cellular metabolism will stop, so it will suffocate the cell, it will stop the metabolic function. So that is when the steep portion come into play. It starts giving out, the hemoglobin starts losing its oxygen and makes it available to the tissue, so that the cellular metabolism can keep on going. Otherwise the whole system will come to collapse. Okay. Another part that is highlighted here, as we can see, is the P50. I'm zooming it out. See P50. So it points out to an area which is written as 25 mmHg. So what is P50 and what is its significance? Now P50, P50, which is again a very important concept. P50, it is the partial pressure at which the hemoglobin is 50% saturated with oxygen. Now, when we are talking about it, we may think that, okay, what we have seen? We have seen that at 100 mmHg, there is 100% saturation. So, obviously, if we calculate, we go by mathematical calculation. So, at a partial pressure of 50, it should be 50% saturated. No, it does not happen like that. It does not happen like that. Why? Because the oxygen and hemoglobin binding follows the sigmoid curve. So, if it was like not following the sigmoid curve, if the oxygen hemoglobin curve was like linear curve, then obviously at 100 mmHg, if the saturation is 100%, then obviously at 50 mmHg, the saturation would have been 50%. But no, this is not happening, because we have just seen when the partial pressure drops from 100 to 75, even then there is no unbinding of the oxygen from the hemoglobin. So P50 is the partial pressure of oxygen in the blood, in which the hemoglobin is 50% saturated. Because of the sigmoid nature, it is seen that it is only like it is about somewhere between 22 to 25 mmHg when there is P50. And P50, another significance is that it is a yardstick. By studying the P50, we can understand how much affinity is there between the oxygen and hemoglobin. So there is a thumb rule to it. All we need to remember, just remember blankly, is that increase is that increase in P50 means decrease in affinity. Whose affinity? Oxygen hemoglobin affinity. So increase, if the P50 value increases, means, say for example, this instead of 25, like for example, instead of 25 mmHg, if P50 increases to 35 mmHg, that means oxygen hemoglobin affinity is decreased, and vice versa. If P50 is at 15 mmHg or say 18 mmHg, which is like less than 22 to 25 range, right? So it means oxygen hemoglobin affinity has increased. This is the significance. So by studying the P50, actually we can understand in what condition the affinity is. Now this is particularly important when the hemoglobin is like, if a person suppose is working in a factory, is working in the traffic, that person is more exposed to emission, and the blood usually get like contaminated in the sense, here substances like a person who's working in the traffic and is not taking the protection is getting constantly subjected to the carbon monoxide emission. So in that person, there is always like binding of carbon monoxide onto the hemoglobin more than the oxygen, because the air that he is inhaling is rich in carbon monoxide. So in this kind of person, the P50 helps to understand the affinity of the oxygen and hemoglobin. This is the take from it. Now, there are certain factors which defines or which helps the oxygen to bind more to the hemoglobin, certain conditions, certain circumstances within the body itself that will help the oxygen to bind more to the hemoglobin, and the opposite is also true. Okay, now these are like explained by the effect which we called as the Bohr effect. So what is Bohr's effect? Now Bohr's effect, it explains the conditions that causes unloading, like coming out of oxygen from the hemoglobin. Now this we understand through a graph. Otherwise, how do we depict it? So I will show you the graph also, let us understand. Say this is like, say for example, this is the sigmoid normal curve. So whenever there is any Bohr's effect, means the oxygen is getting detached from the hemoglobin, this same curve, what it will do, it will shift, it will come below, it will be moved towards right, it will move downward towards right. So this is called as the Bohr's effect. Now what is the importance of Bohr effect? What is the significance? Conditions we will come right away. Now significance of Bohr's effect, what is it significant for? It makes oxygen come out, or the unload, the proper English is unload, from hemoglobin and avail and be available to the tissues. So any condition, if there are certain conditions in which the tissues need oxygen, it is because of the Bohr's effect, because oxygen is like bound to the hemoglobin, right? A saturated hemoglobin also acts as a storehouse of oxygen, right? We can think it that way also, because hemoglobin, it has got like binding site for 8 atoms of oxygen and 4 molecules of oxygen. So if there is a condition that the tissues are suffocating, tissues are going into hypoxia, they need oxygen for their survival, for their metabolism, then where will the oxygen come from? A human being cannot understand the need, right? So the thing is that it is the hemoglobin that will let out its oxygen and give it to the tissues, so that the tissue can carry out its metabolism and it does not go into hypoxic condition. So it is because of the Bohr's effect that the tissues can get oxygen in time of need. Now what are those conditions? Those conditions, those conditions are increase in the metabolite 2,3-diphosphoglyceride. Now, this is a byproduct of glycolysis and it is always happening in the RBC. Okay, so whenever there is increase in the 2,3-DPG, what it will do? It will readily, it will replace the oxygen from hemoglobin and bind to the hemoglobin. So now oxygen comes out of the hemoglobin and it is available for the tissues to use it. This is one condition. Second condition is like increase in temperature. Now suppose during exercise, suppose a person is exercising during exercise. So during exercise, what happens? There is a rapid usage of oxygen. So now the tissues, in an exercising tissue, there is like always need of oxygen. So during exercise, the skeletal muscles need oxygen. So this oxygen, from where will it come? It will come from the hemoglobin. It will come from myoglobin also, we'll come later to that. But first it will come from the hemoglobin. Hemoglobin, when there is increase in the temperature will break the association of the oxygen and hemoglobin, that bond will be broken due to heat, and oxygen will come out of the hemoglobin and it will be provided to the skeletal muscle fibers. This is the second factor. Okay. Third factor is that increase in acidity. Increase in acidity means decrease in pH, that is low pH. So, whenever there is any acid buildup in the body, may it be due to diabetes, may it be due to any other reason, whenever there is any increase in the acidity, that is the pH will go low, it will decrease. Then also there will be oxygen unloading from the hemoglobin. These are the factors that will lead to the oxygen going to the tissue, which is the effect of the Bohr's effect. So, how does this Bohr effect look like? This Bohr effect looks like this. We have already drawn the diagram, so I'll just show you how it looks like in the graph. So, here, this is our Bohr effect diagram. So, this oxygen hemoglobin dissociation curve in Bohr's effect, the oxygen hemoglobin dissociation curve moves right. Okay. And another point when it moves to right is in case of increase in partial pressure of carbon dioxide. Suppose someone has climbed height and there is not enough oxygen and the partial pressure of carbon dioxide is increased, that is hypercapnia. Then also the curve will shift to right. Now here you see, if you see the brown one is our normal oxygen hemoglobin dissociation curve. So if you see very clearly the partial pressure, like it is at 25 where we have the P50 for the normal curve, right? This is our P50. Now the same curve, when under Bohr effect, it moved to right, the blue one, as you can see here distinctly, the P50, it has shifted to more than 25. So here the P50 increased. So due to Bohr's effect, the affinity of hemoglobin and oxygen decreases, right? It decreases and P50 increases. So P50, that was like 22 to 25. Now you see it has become like almost 35. So what we studied is being shown here. Now, this is what happens when there is unloading or Bohr's effect. Now, if the opposite happens, like in opposite conditions, like decrease in 2,3-diphosphoglycerate, decrease in partial pressure of carbon dioxide, decrease in temperature, and increase in pH. That means decrease in H plus concentration, means decrease in acidity. In all this condition, what will happen? In all this condition, the same sigmoid curve, if we draw it like this is the sigmoid curve in normal condition. Now, in all this condition, which is opposite to the Bohr effect, the sigmoid curve will move to left. It will move to left, meaning the affinity of oxygen to hemoglobin increases and P50 decreases. So here you see the P50 will be less and the oxygen to hemoglobin affinity decreases. That means it is the opposite of Bohr effect. There is no term for that. There is nothing like any effect that we attribute to this thing. It is just opposite of Bohr effect. Means when Bohr effect is not working, there is increase in the affinity of oxygen and hemoglobin. Oxygen is not available to the tissue, and oxygen is not coming out of the hemoglobin. This is our Bohr's effect and its condition. Now coming to the Haldane effect, another important concept is the Haldane effect. Now how to understand and remember the Haldane effect? Haldane effect, it actually tells two things. What are the two things? Haldane effect says that unloading of carbon dioxide leads to loading of oxygen to the hemoglobin. Like when carbon dioxide comes away or leaves the hemoglobin, it is letting the oxygen bind to the hemoglobin. Like here you can think it as a opposite of Bohr's effect. But here the emphasis is more on the carbon dioxide. Why more on the carbon dioxide? Because carbon dioxide actually, if the partial pressure of carbon dioxide and oxygen were same in the inspired air, then oxygen would have not gotten a chance to bind to the hemoglobin because its affinity, because carbon dioxide's capacity to bind to the hemoglobin is much more than that of the oxygen. But fortunately, the air that we are inspiring, that air itself has like a lot more of oxygen compared to that of the carbon dioxide. Now, Haldane says that additionally, the event that is helping us to expel or expire carbon dioxide and keep more oxygen inside our system is because carbon dioxide, when it is getting unbound or when it is coming out of the hemoglobin, it is actually aiding or helping oxygen to bind more to the hemoglobin. It is saying the loading of the oxygen, but in terms of the carbon dioxide. So, we can say that every time, in a very simple way, if we have to remember, okay, every time oxygen binds to the hemoglobin, carbon dioxide is let out or expired via the lungs. And each time oxygen is let out, or rather say oxygen is available to the tissue, meaning what? The oxygen comes out of the hemoglobin and is available to the tissue. Carbon dioxide binds to the hemoglobin. This is the important concept. Means when oxygen is binding to the hemoglobin, carbon dioxide is getting out of the body through the lungs, through expiration. So this is happening every time oxygen is binding to the hemoglobin. Similarly, each time oxygen is coming out of the hemoglobin and going to the tissues for its function, that time when the oxygen is leaving the hemoglobin, carbon dioxide is coming and binding onto the hemoglobin. This is what the Haldane says. And it is because of the Haldane effect that we are able to expire more volume of carbon dioxide than we should be able to. So we understand this one with this graph, which is the Haldane effect. So this is the graph which explains the Haldane effect. So here we see 3 points. The 3 points are point A, point A, point B and point C. Now what at this point? First of all, let's remember point A is at tissues. How do we understand? How do we remember it is a tissue? Here you see partial pressure is 40 mmHg. So where is partial pressure of oxygen in 40 mmHg? It is in the tissues, in the venous blood, right? So it is happening at the tissue. And here you see on the x-axis, it is the partial pressure of the carbon dioxide. The partial pressure of the carbon dioxide at the tissue is like 45. So this A event is happening at the tissues. Now, point B and C are at lungs. So here you see B at the partial pressure of carbon dioxide is like 40, which is at the lungs. And C also it is 40, which is at the lungs. Now here, what does Haldane effect says that point A, that is at tissues level. Okay. The partial pressure of oxygen is 40 mmHg. The partial pressure of oxygen is 40 mmHg. Now, if it is parallel, if it is matched to the point on the y-axis. Now, y-axis is the carbon dioxide concentration, volume in ml in the blood, which is reflecting the percentage, and these values are given in the percentage form. So, point A, if we go by the dotted lines, it is corresponding, which is at the level of the tissue, it is corresponding to 52% of the carbon dioxide in the venous blood at the level of the tissue. So, when the partial pressure of oxygen is 40 mmHg, where in the tissue, the carbon dioxide percentage is 52%. It is as high as the 52%. Okay. After that, now think of the point C. First, we will think of the point C. Later, we will come to B. So, if at the level of the tissue, that is at point C. The point C, what does it show? It shows absence of Haldane effect. So, when this venous blood at a partial pressure of like 40 comes to the lungs, this B and C is at the lung. So, in the C, it is in the lungs. When the partial pressure of carbon dioxide at the lungs is 40, its percentage in the lung is only 50%. Means from the tissue, when it came to the lungs, in absence of Haldane effect, the partial pressure of carbon dioxide dropped from 52% to 50%. That is in the absence of the Haldane effect. So, in absence of Haldane effect, only 2% of carbon dioxide got expired. only 2% of carbon dioxide got expired. From 52, it has become 50. Means from the tissue, that 52% of carbon dioxide, when it came to the lungs, in absence of Haldane effect, only 2% got expired out. So if 2% got expired out, means only 2% of oxygen has the space to bind, because 2% carbon dioxide expired out means where did it expire out from? It was on the hemoglobin. Now from the hemoglobin, it got unbound and it went to the atmosphere via expiration. So in absence of Haldane effect, from 52 only till 50, only 2% of carbon dioxide we could expire to the atmosphere, and that equivalent, that is 2% of oxygen, now has the capacity to bind in the arterial blood, okay, at the level of the lungs. Now here, this is our point C. Now point C, this is also happening, this is happening at the lungs. The point C is also happening at the lungs in presence of Haldane effect. Now in presence of Haldane effect, what do we see? We see the partial pressure of carbon dioxide dropped from, like, the partial pressure rather expressed in content dropped from like 52% to 48%, that is partial pressure in content form, to 48%. So 52 minus 48, that is 4% of carbon dioxide got expired. So in presence of Haldane effect, if we look again, now point B, which is also happening at the level of the lungs, because here the partial pressure of carbon dioxide is 40. So the content drop, here we can see from 52, it has dropped all the way to 48. That means in presence of Haldane effect, the unbinding, the expiration, the loss of carbon dioxide content is double than what it would have happened in absence of Haldane effect. So its indirect implication is what? In presence of Haldane effect, the oxygen has more chance, double the chance to bind to the hemoglobin compared to what would have happened if the Haldane effect was absent. So this is our Haldane effect. This is our understanding of the Haldane effect. This is the content. Okay. So why Haldane effect is important? Because Haldane effect, via the carbon dioxide, it explains the loading or binding of oxygen to the hemoglobin. Okay. So this was our topic today related to the respiratory membrane and the oxygen hemoglobin dissociation curve, and what was its content, how to calculate the content, the Fick's law, its implication, and lastly we understood the Bohr's effect and the Haldane effect. So in the next lecture we will complete the remaining part of the respiratory system and move on to the next system. So till then, thank you.